Good day. For this video, I'm going to discuss about the water pollution. The uses we make of water in lakes, rivers, ponds, and streams is greatly influenced by the quality of the water found in them. Activities such as fishing, swimming, boating, shipping, and waste disposal have very different requirements for water quality. In many parts of the world, the introduction of pollutants from human activity has seriously degraded water quality, even to the extent of turning pristine trout streams into foul open sewers with few life forms and fewer beneficial uses. So in this topic, I'm going to discuss about the pollutants and their sources. First is the point sources, non-point sources, oxygen-demanding material, nutrients, pathogenic organisms, suspended solids, salts, pharmaceuticals and personal care products, endocrine disrupting chemicals, pesticides, and heat. These are the sources of water pollution. Point sources. Domestic sewage and industrial wastes are called point sources because they are generally collected by a network of pipes or channels and conveyed to a single point of discharge into receiving water. Domestic sewage consists of wastes from home, schools, office buildings, and stores. The term municipal sewage is used to mean domestic sewage into which industrial wastes are also discharged. Non-point sources. Urban and agricultural runoff are characterized by multiple discharge points. These are called non-point sources. Often the polluted water flows over the surface of the land or along natural drainage channels to the nearest water body. Much of the non-point sources pollution occurs during the rainstorms or spring snowmelt, resulting in large flow rates that make treatment even more difficult. Runoff from agricultural land is a significant non-point source. Fertilizers, whether in the form of manure or commercial fertilizer, contributes nutrients. Agricultural runoff carries nutrients and toxic organic compounds. Soil erosion contributes also suspended solids. Oxygen-demanding material. Anything that can be oxidized in the water receiving with the consumption of the salt, molecular oxygen, is termed oxygen-demanding material. This material is usually biodegradable organic matter but also includes certain organic compounds. The consumption of dissolved oxygen or DO poses a threat to fish and other higher forms of aquatic life that must have oxygen to live. The critical level of DO varies greatly among species. Almost any natural occurring organic matter such as animal droppings, crop residues, or leaves that get into the water from non-point sources contribute to the depletion of DO. Nutrients. Nitrogen and phosphorus, two nutrients of primary concerns, are considered pollutants because they are too much of a good thing. All living things requires these nutrients for growth. Thus, they must be present in rivers and lakes to support the natural food chain. Pathogenic organisms. Microorganisms found in wastewater include bacteria, viruses, and protozoa excreted by diseased person or animal. When discharged into surface waters, they make water unfit for drinking, that is, non-potable. Cholera and typhoid are endemic diseases in the world with over 384 cases of cholera and 16 million cases of typhoid per year. These killers cause 20,000 and 600,000 deaths per year respectively, according to the Population Reports 1998. Suspended solids. Organic and inorganic particles that are carried by the wastewater into receiving water are termed suspended solids. When the speed of the water is reduced by flowing into a pool or a lake, many of these particles settle to the bottom or as sediments. Organic suspended solids may also exert an oxygen demand. Inorganic suspended solids are discharged by some industries but result mostly from soil erosion, which is particularly bad in areas of logging, strip mining, and construction activity, as excessive sediment loads are deposited into lakes and reservoirs. The usefulness of water is reduced. Salts. Although most people associate salty water with oceans and salt lakes, all water contains some salt. These salts are often measured by evaporation of a filter water sample. The salts and other things that don't evaporate are called total dissolved solids or TDS. A problem arises when the salt concentration is normally fresh water increases to the point where the natural population of plants and animals is threatened or the water is no longer useful for public water supplies or irrigation. High concentrations of salt are discharged by many industries, and the use of salt on roads during winter causes high salt levels in the urban runoff, especially during the spring snowmelt. Pharmaceuticals and personal care products or PPCPs. These are the class of compounds that are applied externally or ingested by humans, pets, and other domesticated animals. They are released to the environment through the disposal of expired, unwanted, or excess medications to the sewage system by individuals, pharmacies, or physicians. Endocrine disrupting chemicals. The class of chemicals known as endocrine disruptors or EDCs alters the normal physiological function of the endocrine system and can affect the synthesis of hormones. Some natural occurring endocrine disrupting chemicals such as 7-B estradiol that is both a natural occurring estrogen and a synthetic estrogen used as method of birth control are excreted into wastewater. Other EDCs include the polychlorinated biphenyls, commonly used pesticides as atrazine and triazine chemicals in the plantlates. Pesticides. The major classes of pesticides are herbicides, insecticides, and fungicides. Herbicides are used to kill weeds. The most commonly used herbicides are astrazine, metolachlor, and alachlor. Chlorpyrifos and diazinon and malathion are the typical insecticides used to kill insects that would otherwise destroy crops, gardens, or structures. Common fungicides are sulfur, chlorothalonil, and mancozeb. Heat. Although heat is not often recognized as pollutant, those in the electric power industries are well aware of the problems of disposing of waste heat. Also, waters released many industrial processes are much hotter than the receiving waters. In some environments, an increase of water temperature can be beneficial. For example, production of clams and oysters can be increased in some areas by warming the water. On the other hand, increases in water temperature can have negative impacts. Many import commercials and game fish, such as salmon and trout, live only in cool water. In some instances, the discharge of heat water from a power plant can completely block salmon migration. Higher temperatures also increase the rate of oxygen depletion in areas where oxygen demanding wastes are present. So, pollution in rivers includes total maximum daily load or TMDL, effect of oxygen demanding wastes on river, biochemical oxygen demand, graphical determination of BOD constants, laboratory measurement of biochemical oxygen demand, additional notes on biochemical oxygen demand, nitrogen oxidation, Streeter Phelps DO SAG curve, effect of nutrients on water quality in rivers. Total maximum daily load or TMDL under section 303D of the 1972 Clean Water Act states territories and authorized tribes are required to develop lists of impacted waters. Impaired waters are those that do not meet water quality standards that the state territories and authorized tribes have established for them. This assessment is made after assuming that point sources of pollution have installed minimum levels of pollution control technology. Total maximum daily load or TMDL. The law requires that these jurisdictions establish priority rankings for waters on the list and develop total maximum daily loads or TMDL. For these waters, a TMDL specifies the maximum amount of pollutant that a water body can receive and still meet water quality standards. The TMDL computation is defined as total maximum daily load is equal to summation of WLA plus summation of LA plus MOS, where WLA waste load allocations, that is portions of the TMDL assigned to existing and future point sources. LA is load allocation, that is portion of the total maximum daily load assigned to existing and future non-point sources. And MOS is the margin of safety. Let's proceed to effect of oxygen demanding waste demanding wastes on river. The introduction of oxygen demanding material, either organic or inorganic, into a river causes depletion of dissolved oxygen in the water. This poses a threat to fish and other higher forms of aquatic life if the concentration of oxygen falls below a critical point. Organic oxygen demanding materials are commonly measured by determining the amount of oxygen consumed during degradation in a manner approximating degradation in natural waters. This section begins by considering the factors affecting oxygen consumption during the degradation of organic matter, then moves on to inorganic nitrogen oxidation. Biochemical oxygen demand. First is the theoretical oxygen demand, or THOD. The amount of oxygen required to oxidize a substance to carbon dioxide and water may be calculated by stoichiometry if the chemical composition of the substance is known. This amount of oxygen is known as the theoretical oxygen demand, or THOD. Example, compute the TOHD of 108.75 mg of per liter of glucose. In solution, we begin by writing a balanced equation for the reaction. Next, compute the gram molecular weights of the reactants. Chemical oxygen demand, or COD. In contrast to the TOD, the chemical oxygen demand, pronounced COD, is measured quantity that those that depend on knowledge of the chemical composition of the substances into water. In the COD test, a strong chemical oxidizing agent, or chromic acid, is mixed with a water sample then boiled. The difference between the amount of oxidizing agent at the beginning of the test and that remaining at the end of the test is used to calculate the COD. Biochemical oxygen demand, or BOD. If the oxidation of the organic compound is carried out by microorganisms using the organic matter as a food source, the oxygen consumed is known as biochemical oxygen demand. The actual biochemical oxygen demand is less than the theoretical oxygen demand due to the incorporation of some of the carbons into new bacteria cells. The BOD test is an indirect measurement of organic matter because we actually measure only the change in dissolved oxygen concentration caused by the microorganisms as they degrade the organic matter. BOD rate equation is equal to LO times 1 minus 10 minus KT, where LO is oxygen equivalent of organic compounds at time equals 0. K is used for constant in base 10 and K reaction base constant in base E. K equals 2.303 K. Proceed to graphical determination of BOD constants. A variety of methods may be used to determine K and LO from experimental set of BOD data. The simplest and least accurate method is to plot BOD versus time. The rate equation is used to solve for K. One simple method around this is called Thomas graphical method. Thomas graphical method procedures for determining the BOD constants. From the experimental results of BOD for various values of T, calculate time over BOD time 1/3 for each day using the equation below. Next is plot time over BOD time 1/3 versus T time on an arithmetic graph paper and draw the line of best fit by eye. Third, determine the intercept A and B slope from the plot. Lastly, calculate K and LO. Using LO is equals to 1 over 6 times A squared times B. Laboratory measurement of biochemical oxygen demand. In order to have as much consistency as possible, it is important to standardize testing procedures when measuring BOD. For the next slide, standard BOD test is outlined with emphasis placed on the reason for each step. So these are the steps. Sample given. A special 300 ml BOD bottle is completely filled with a sample of water that has been approximately diluted and inoculated with microorganisms. The bottle is then stoppered to exclude air bubbles. The most oxygen that can be dissolved is about 9 mg per liter, so the BOD of the diluted sample should be between 2 and 6 mg per liter. The dilution water also contains an inoculum of microorganisms, so that all samples tested on a given day contain approximately the same type and number of microorganisms. The ratio of undiluted to diluted sample is called the sample size, usually expressed as a percentage, while the inverse relationship is called the dilution factor. Mathematically, these are the sample size in percent is equal to volume of undiluted sample over volume of diluted sample times 100. Dilution factor is equal to the volume of diluted sample over volume of undiluted sample is equal to 100 over sample size. The appropriate sample size is used can be determined by dividing 4 mg per liter by the estimated BOD concentration in the sample being tested. A convenient volume of undiluted sample is then chosen to approximate to this sample size. Step 2. Blank samples contain only the inoculated dilution water are also placed in BOD bottles and stoppered. Blanks are required to estimate the amount of oxygen consumed by the added inoculum of microorganisms called seed in the absence of the sample. Step 3. The stoppered BOD bottles contain diluted samples and blanks are inoculated in the dark at 20 degrees Celsius for desired number of 4 days. For most purposes, a standard time of 5 days is used. To determine the ultimate BOD and the BOD rate constant, additional times are used. As mentioned earlier, the BOD test is conducted at the standard temperature of 20 degrees Celsius so that the effect of temperature on the BOD rate constant is eliminated and results from different laboratories can be compared. Step 4. After the desired number of days has elapsed, the samples and blanks are removed from the incubator and the dissolved oxygen concentration in each bottle is measured. The BOD of the undiluted sample is then calculated using the following equation below. So let's proceed to the additional notes on biochemical oxygen demand. Although the 5-day BOD have been chosen as the standard value for most wastewater analysis and for regulatory purposes, ultimate BOD is actually a better indicator of total waste strength. For any one type of waste having a defined BOD rate constant, the ratio between the ultimate BOD and the BOD5 is constant so that BOD5 indicates relative waste strength. For different types of waste having the same BOD5, the ultimate BOD is the same only if, by chance, the BOD rate constants are the same. The 5-day BOD was chosen as the standard value for the most purposes because the test was devised by environmental engineers in England, where rivers have travel times to the sea of less than 5 days. So there was no need to consider oxygen demands at longer times. Since there is no other time that is any more rational than 5 days, this value has become firmly entrenched. This is illustrated in the figure below for municipal wastewater having a K equals 0.34 d raised to the power of -1 and industrial wastewater having a K is equals 0.115 d raised to the power of -1. So the effect of K on ultimate BOD for wastewater having the same BOD. The effect of K on BOD5 when the ultimate BOD is constant. So in laboratory and in river temperature. So let's proceed to nitrogen oxidation. Many organic compounds such as proteins also contains nitrogen that can be oxidized when the consumption of molecular oxygen. Two processes must be considered separately because the mechanisms and rates of nitrogen oxidation are distinctly different from those of carbon oxidation. The logically, oxygen consumption due to oxidation of carbon is called carbonaceous BOD or CBOD, while that due to nitrogen oxidation is called nitrogenous BOD. The nitrogen is released into the surrounding water as ammonia or NH3 at normal power of hydrogen values. This ammonia is actually in the form of ammonium cation. The ammonia released from organic compounds plus that from other sources such as industrial wastes and agricultural runoff is oxidized to nitrate by a special group of nitrifying bacteria as their source of energy in a process called nitrification. The overall reaction for ammonia oxidation is this formula or this equation. From this reaction, the theoretical NBOD can be calculated as follows. Gram is equal to grams of oxygen used over grams of nitrogen oxidized. The rate at which the NBOD is exerted depends heavily on the number of nitrifying organisms present. In untreated sewage, there are few of these organisms, while in a well-treated effluent, the concentration is high. When samples of untreated and treated sewage are subjected to the BOD test. In case of untreated sewage, the NBOD is exerted after much of the CBOD or carbon biochemical oxygen demand has been exerted. In the case of the treated sewage, a higher population of nitrifying organisms in the sample reduces the lag time. When measurement of only carbonaceous or BOD is desired, chemical inhibitors are added to stop the nitrification process. The rate constant for nitrification is also affected by temperature. Street Phelps Dussault curve. One of the major tools of water quality management in rivers is the ability to assess the capability of a stream to absorb a waste load. This is done by determining the profile of DO concentration downstreams from a waste discharge. This profile is called the DO sag curve because the DO concentration dips as oxygen demanding materials are oxidized and then rises again further downstreams as the oxygen is replenished from the atmosphere. Develop a mathematical expression for the DO-SAG curve. The sources of oxygen and the factors affecting oxygen depletion must be identified and quantified. The only significant sources of oxygen are radiation from the atmosphere and photosynthesis of aquatic plants. Oxygen depletion is caused by a larger range of factors, the most important being the BOD, both carbonaceous and nitrogenous of the waste discharge, the BOD already in the river upstream of the waste discharge. The second most important factor is that the DO in the discharge is usually less than that in the river. Thus, the DO at the river is lowered as soon as the waste is added, even before any BOD is exerted. Other factors affecting dissolved oxygen depletion includes non-point source pollution, the respiration of organisms living in the sediments, and the respiration of the aquatic plants. So these are the qualitative discussing about the other factors affecting DO levels. Effects of nutrients on water quality in rivers. Although oxygen demanding wastes are the most important river pollutants on the an overall basis, nutrients can also contribute to deteriorating rating water quality in rivers by causing excessive plant growth. Some plant is desirable since plant form of the base of the food chain and thus support the animal community. However, excessive plant growth can create a number of undesirable conditions such as thick slime layers on rocks and dense growth of aquatic weeds. Effects of nitrogen. There are three reasons why nitrogen is detrimental to a receiving body. First is in high concentration of NH3, nitrogen is toxic to fish. So ammonium is in low concentrations and NO3 serves as nutrients for excessive growth of algae. Conversion of NH4 plus to NO2 consumes large quantities of dissolved oxygen. Effects of phosphorus. The major deleterious effect of phosphorus is that it serves as a vital nutrient for the growth of algae. If the phosphorus availability meets the growth demand of the algae, there is an excessive production of algae. When algae die, they become an oxygen-demanding organic material as bacteria seek to degrade them. This oxygen demand frequently overtaxes the DO supply of the water body and as consequence causes fish to die. So let's proceed now to water pollution in lakes, certification and turnover, biological zones, lake productivity, eutrophication, algae growth requirements, delimiting nutrient, control of phosphorus in lakes, acidification of lakes. Stratification and turnover. Nearly all lakes in the temperate climate zones become stratified during the summer and overturn in the fall due to changes in the water temperature that results from the annual cycle of air temperature changes. In addition, lakes in cold climates undergo winter stratification and spring overturn as well. So in summer, during summer, the surface water of a lake is heated both indirectly by contact with warm and directly by sunlight. Warm water, being less dense than cool water, remains near the surface until mixed downward by turbulence, wind, waves, both, and other forces. In summer, because this turbulence extends only a limited distance below the water surface, the result is an upper layer of well-mixed warm water, the ephelimnium, floating on the lower water, the hypolimnium, which is poorly mixed and cool, as shown in the figure. Because of good mixing, an ephelimnium will be aerobic. The hypolimnium will have a lower DO and may become anaerobic, devoid of oxygen. In the fall, as temperatures drop, the ephelimnium cools until it is more dense than the hypolimnium. The surface water then sinks, causing overturning. The water of the hypolimnium rises to the surface where it cools and again sinks. The lake thus becomes completely mixed. In winter, if the lake is in a cold climate, this process stops when the temperature reaches 4 degrees Celsius, since this is the temperature at which water is most dense. Further cooling or freezing of the surface water results in winter stratification, as shown in the second figure. Spring. As the water warms in the spring, it again overturns and becomes completely mixed. Thus, temperature climate lakes have at least one, if not two, cycles of stratification and turnover every year. Biological zones. Lake contains several distinct zones of biological activity, largely determined by the availability of light and oxygen. The most important biological zones are the euphotic, littoral, and benthic zones. Most important biological zones, the benthic zones. Euphotic zones, profound zones below euphotic zone, and lateral zone. Euphotic zone, the upper layer of water which sunlight can penetrate, is called the euphotic zone. All plants growth occurs in this zone. In deep water, algae are the most important plants, while rooted plants grow in shallow water near the shore. The upper layer of the water which sunlight can penetrate is called the euphotic zone. In the euphotic zone, plants produce more oxygen by photosynthesis than they remove by respiration. Below the euphotic zone lies the profundal zone. The transition between the two zones is called the light compensation level. It corresponds roughly to a depth at which the light intensity is about 1% of unattenuated sunlight. The littoral zone. The shallow water near the shore in which rooted water plants can grow is called littoral zone. The extent of the littoral zone depends on the slope of the lake bottom and the depth of the euphotic zone. The littoral zone cannot extend deeper than the euphotic zone. Benetic zone. The bottom sediments comprises the benetic zone. As organisms living in the overlying water die, they settle to the bottom where they are decomposed by organisms living in the benetic zone. Bacteria are always present. Lake productivity. The productivity of a lake is a measure of its ability to support a food web. Algae from the base of this food web supplying food for the higher organisms. The lake's productivity may be determined by measuring the amount of algal growth that can be supported by the available nutrients. Although a more productive lake usually will have a higher full fish population, number of the most desirable fish may decline. In fact, increased productivity generally results in reduced water quality because of undesirable changes that occur as algal growth increases. Because of the important role productivity plays in determining water quality, it forms a basis for classifying lakes. Lake classification based on productivity. Oligotrophic lakes have a low level of productivity due to a severely limited supply of nutrients to support algal growth. As a result, the water is clear enough that the bottom can be seen at considerable depths. In this case, the euphotic zone often extends to the hypolynium, which is aerobic. Oligotrophic lakes therefore support cold water game fish. Lake Tahoe in the California-Nevada border is a classic example of oligotrophic lake. Eutrophic lakes have a high productivity because of an abundant supply of algal nutrients. The algae cause the water to be highly turbid, so the euphotic zone may extend only partially into the epithelium. As the algae die, they settle to the lake bottom where they are decomposed by benthic organisms. In eutrophic lake, this decomposition is sufficient to deplete the hypolymium of oxygen during summer certification. Because the hypolymium is anaerobic during the summer, eutrophic lake supports only warm water fish. In fact, most cold water fish are driven out of the lake long before the hypolymium becomes anaerobic because they generally require dissolved oxygen levels of at least 5 milligrams per liter. Highly eutrophic lakes may also have large mats of floating algae that typically impart unpleasant taste and odor to the water. Mesotrophic lakes. These are lakes which are intermediate between oligotrophic and eutrophic are called mesotrophic. Although substantial depletion of oxygen may have occurred in the hypolymium, it remains aerobic. Senescent lakes. These are very old, shallow lakes which have thick organic sediments and rooted water plants in great abundance. These lakes will eventually become marshes. Eutrophication is a natural process in which lakes gradually become shallower and more productive through the introduction and cycling of nutrients. Thus, oligotrophic lakes gradually pass through the mesotrophic, eutrophic, and senescent stages, eventually filling completely. The time for this process to occur depends on the original size of the lake and rate at which sediments and nutrients are introduced. In some lakes, an eutrophication process is so slow that thousands of years may pass with little change in water quality. Other lakes may have been eutrophic from the day they were formed if nutrients levels were high at that time. Cultural eutrophication is caused when human activity speeds the processes naturally occurring by increasing the rate of which sediments and nutrients are added to the lake. Thus, lake pollution can be seen as an intensification of natural process. This is not to say that particular lakes are necessarily polluted, but that pollution contributes to eutrophication. Algae growth requirements. All algae requires macronutrients such as carbon, nitrogen, and phosphorus, and micronutrients such as trace elements. For algae to grow, all nutrients must be available. Lack of any one nutrient will limit the total algae population. The availability of each nutrient in each natural cycle are summarized below. Carbon. Algae obtain their carbon from carbon dioxide dissolved in the water. Because the carbon dioxide is in equilibrium with the bicarbonate buffer system, the immediate available carbon is determined by the alkalinity of the water. However, as carbon dioxide is removed from the water, it is replenished from the atmosphere. The atmosphere is, of course, a virtually inexhaustible source of this gas. When algae are either consumed by higher organisms or die and decompose, the organic carbon is oxidized back to carbon dioxide, which returns either to the water or to the atmosphere to complete the carbon cycle. Nitrogen. Nitrogen cycles from nitrate to organic nitrogen to ammonia and back to nitrate as long as the water remains aerobic. However, in anaerobic sediments and in the hypolynium of eutrophic lakes, when algae decomposition has depleted the oxygen supply, nitrate is reduced by anaerobic bacteria to nitrogen gas and lost from the system in a process called denitrification. Denitrification reduces the average time nitrogen remains in lake system. The denitrification reaction is 2NO3 plus organic carbon yields NO2 plus CO2 plus H2O. Some photosynthetic microorganisms can also fix nitrogen gas from the atmosphere by converting it into organic nitrogen. In lakes, the most important nitrogen-fixing microorganisms are photosynthetic bacteria called cyanobacteria, formerly known as blue-green algae because of the pigments they contain. Phosphorus. Lace originates from external sources and is taken up by algae in the organic form and incorporated into organic compounds. During algal decomposition, phosphorus is returned to the inorganic form. The release of phosphorus from dead algae cells is so rapid that only a little of its leaves and epithelium will with the settling algae cells. However, little by little, phosphorus is transferred to the sediments, some of it in uncomposed organic matter, some of it in precipitates of iron, aluminum, and calcium, and some bound to clay particles. Trace elements. The quantities of trace elements required to support algal growth are so small that most fresh water have sufficient amount of substantial algae population. The limiting nutrient. In 1940, Justin Liebig formulated the idea that growth of a plant is dependent on the amount of foodstuff that is presented to it in minimum quantity. This is now known as Liebig's law of the minimum. As applied to algae, it means that algal growth will be limited by the nutrients that is least available. Of all the nutrients, only phosphorus is not readily available from the atmosphere or natural water supply. For this reason, phosphorus is deemed the limiting nutrient in lakes. The amount of phosphorus controls the quantity of algal growth and therefore the productivity of lakes. This can be seen from the figure in the next slide, in which concentration of chlorophyll is plotted against phosphorus concentration. So relationship between summer levels of chlorophyll and measured total phosphorus concentration for 140 lakes. Chlorophyll A, one of the green pigments involved in photosynthesis, is found in all algae, so it is used to distinguish the amount of algae in the water from other organic solids such as bacteria. It has been estimated that the phosphorus concentration should be below 0.010 to 0.15 milligrams per liter to limit algae blooms. Control of phosphorus in lakes. Because phosphorus is usually the limiting nutrient, control of cultural eutrophication must be accomplished by reducing the input of phosphorus to the lake. Once the input is reduced, the phosphorus concentration will gradually fall as phosphorus and is buried in the sediment or flushed from the lake. In conjunction with reduced phosphorus inputs, these measures can help speed up the removal of phosphorus already in lake system. Of course, the need to speed the recovery process must be weighed against the potential damage from inundating shoreline areas with sludge and stirring up toxic compounds buried in the sediment. Water To be able to reduce phosphorus inputs, it is necessary to know the sources of phosphorus and the potential of their reduction. The natural source of phosphorus is the weathering of rock. Phosphorus released from the rock can enter the water directly, but more commonly it is taken up by plants and enters the water in the form of dead plant matter. It is exceedingly difficult to reduce the natural inputs of phosphorus. If these sources are large, the lake is generally naturally eutrophic. Municipal and industrial wastewaters. All municipal sewage contains phosphorus from human excrement, meaning industrial waste are high in this nutrient. In these cases, the only effective way of reducing phosphorus is through advanced waste treatment processes. Municipal wastewaters also contains large quantities of phosphorus from the detergents containing polyphosphate, which is a chain of phosphate ions linked together. The polyphosphate binds with a hardness in water to make the detergent a more effective cleaning agent. Septic tank seepage. The shore of many lakes are dotted with homes and summer cottages, each with its own septic tank and a tile field of waste disposal. As treated wastewater moves through the soil toward the lake, phosphorus is absorbed by the soil particles, especially clay. Thus, during the early life of the tile field, very little phosphorus gets to the lake. However, with time, the capacity of the soil to absorb phosphorus is exceeded and any additional phosphorus will pass into the lake, contributing to eutrophication. The time it takes for phosphorus to break through to the lake depends on the type of soil, the distance of the lake, the amount of wastewater generated, and the concentration of phosphorus in that wastewater. To prevent phosphorus from reaching the lake, it is necessary to put the tile field far enough from the lake that the absorption capacity of the soil is not exceeded. If it is not possible, it may be necessary to replace the septic tanks and tile fields with a sewer to collect the wastewater and transport it to a treatment facility. Agricultural runoff. Because phosphorus is a plant nutrient, it is an important ingredient in fertilizers. As rainwater washes off fertilized fields, some of the phosphorus is carried into streams and then into lakes. Most of the phosphorus not taken up by growing plants is bound to soil particles. Bound phosphorus is carried into streams and lakes through soil erosion. Acidification of lakes. Most lakes are buffered by the carbonate buffer system to the extent that the buffer capacity of the lake is not exceeded. The pH of the lake will not be appreciably affected by acid rain. If there is a source of carbonate to replace the consumed by the acid rain, the buffering capacity can be quite large. Calcareous soils are those containing large quantities of calcium and carbonate. Most lakes are buffered by the carbonate buffer system to the extent that the buffer capacity of the lake is not exceeded. The beach of the lake will not be appreciably affected by acid rain. Estuaries. An estuary is formed along the coastline where the fresh water from rivers and streams flow into ocean. It is a place of transition where fresh water mixes with salt waters. Estuaries are influenced by tides. The mix of salt water and fresh water combined with the diurnal cycle of tidal wetting and drying and changes in salinity make the estuary ecosystem extremely complex. Numerous different habitats supports an abundance of a wide variety of different animals. Which of these is sensitive to minor changes in quality of the water? Like other surface waters, estuaries are subject to a plethora of pollutants, although the technologies and policies that serve to manage the water quality of lakes and rivers also apply to estuaries. The management of the water quality is complicated by the number of political jurisdictions that touch on the estuaries as well as the competing needs of organisms that live there. Groundwater pollution, uncontrolled release, saltwater intrusion. Groundwater, by its very location, has a large measure of protection from contaminants that are found in surface waters. However, once groundwater becomes contaminated, this location and low rate of replacement with fresh water makes it difficult to return it to a pristine state. Two major sources of contaminants are of concern: uncontrolled release of biological and chemical contaminants and saltwater intrusion from overpumping of wells. Uncontrolled releases comes from a variety of sources. Discharge from improperly operated or located septic systems, leaking underground storage tanks, improper disposal of hazardous or other chemical wastes, spill from pipelines or transportation accidents, recharge of groundwater with contaminated surface water, leaking landfills, leaking retention ponds or lagoons, dissolved contaminants plume. Here is the diagram. The properties of the contaminant in the aquifer material govern the migration of the contaminants. Water-soluble chemicals are likely to move vertical down through the soil to the aquifer and migrate with the water as shown in the figure. Saltwater intrusion. Freshwater aquifer, aquifers near ocean or above saline aquifers may become contaminated with salt water when water is pumped from wells and that draw water that is too close to the freshwater saltwater interface. Because seawater is heavier than freshwater, it will form a saltwater wedge in aquifers that drain into the ocean. End of slides. Thank you.